Okay, today we're going to talk about pancreatic hormones and metabolic regulation. In other words, we're talking about those hormones that will control metabolism. And I will separate this lecture into 3 videos. And then we are going into the 5th week of Molecular Endocrinology, BCG 4130. When we talk about hormones, we always consider the hypothalamus, pituitary gland, and then the target organs and the linkages among all these different regulatory mechanisms controlling hormones. And I put this up here mainly because the missing link of how brain controls blood sugar and insulin is still not clearly understood. We only understood exactly 100 years ago that a hormone from the pancreas called insulin could regulate blood sugar. Okay, so that would come to the conclusion that, we may provide the patient with insulin and that would cure a disease called diabetes mellitus or DM. And since then, we focus on the pancreas, and then not much is known, and then we are now studying a lot on how the brain controls sugar and insulin. And let's start with these different videos by doing video 1 and 2 on feeding, fasting, and pancreatic hormones. And we also talk about insulin in the first video. The second video will focus on another hormone counteracting the effects of insulin known as glucagon. Glucagon. Glu means glucose. Glucagon, to add sugar. Okay? So counteracting the effects of insulin. Insulin is from its name the factors from islet gland or from the islet. So it's called insulin, that is to promote sugar uptake, or in other words, would reduce blood sugar level. And this one is to promote blood sugar level. So we'll talk about that, and together with diabetes mellitus in video 2. And then video 3, we're focusing on fatty acid synthesis, and there are also several different hormones that are regulating fatty acid synthesis. Before we start this first video, please pay attention to the other 2 videos known as hormonal control of lipid metabolism and also hormonal control of sugar metabolism that we did 3-4 years ago with Dr. Patrick I. And so before reading all these videos or this slide content, if you do not know much about sugar metabolism or lipid metabolism, please watch these videos first. I've also put these videos up in my YouTube channels and also in the blackboard site of this course. Okay, so I'm not going to show this to you. So please watch them beforehand. Okay, but for the metabolism is concerned, here we only or mainly focus on sugar, and then we put sugar and lipid together because these are all the major carbon that we use every day. Sugar, we use it as energy. However, we have to convert them all into glucose. It's a 6-carbon sugar. Okay. Lipid is another form, not carbohydrate, but lipid is the carbon molecules with a polymer forming into fatty acids. Okay, so these are all carbon materials that we could store, like fatty acid, we could store, or fats, we can store inside our body. So if we want to utilize them, we have to convert fatty acids or fats back into sugar. So they are closely linked together. Okay, so at the end we have this metabolic control of fatty acids in video 3. Okay, so let's start with, sorry, and then we stop here. There's another one, also the hormonal control of sugar metabolism. All right, and so, yeah, feeding and fasting. Number one, when we feed on our food, we consume the food and make them become sugar. So after a big meal with glucose surge, so increase of sugar, insulin, the hormone come from the beta cell in the pancreas, is released to promote glucose uptake because we need all this glucose in our body. We don't need them in the blood. We do need them. We do need them in the blood to transfer to different parts of the body, to the muscles, the tissues, and also to adipose tissue for fat storage. And then in the liver, the sugar will be stored in the form of what we call its glycogen. So it's a polymer of glucose 1-phosphate. Insulin also facilitates, of course, glucose uptake. It would induce protein synthesis and also induce or promote lipid formation. Because after all, if we don't use those sugar, we'll store them as lipid. And then we also need to control this, what we call nutrient flow from the intestine. So when all these materials being digested and then diminishes out, at the stage we call this fasting. The blood glucose and insulin would drop significantly or drop to normal, depends on what level you're talking about. And then at the same time, if it is below normal, another hormone known as glucagon. Glu is glucose, and then glucagon, that is to enhance glucose to be produced. So that hormone is released to prevent hypoglycemia, low sugar or low glucose in blood called hypoglycemia. By promoting glucose production, remember that it's a sugar, it could come from new synthesis, we call it glycogenolysis. That is to degrade glycogen and make them into sugar. Or we may make some new sugar, we call it gluconeogenesis. New glucose being made, okay? There are two pathways that make sure that we have enough energy or glucose energy to survive. And this production of glucose mainly from the liver. Finally, the interaction or interplay between insulin and glucagon. Insulin can depress glucagon in our cells. And same thing is true, glucagon will counteract the effects of insulin. And they have opposite or opposing effects on blood glucose levels. We have a full chart for you in the next slide. So during fasting, we need more sugar. When we are well fed, we have a lot of sugar. So this sugar, we start from the right hand side here. We start from here. You can see that would induce insulin. So insulin would promote the metabolism of sugar. And this metabolism of sugar, including turning glucose into glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate, and then phosphoenolpyruvate or PEP. And then we go into glycolysis. And glycolysis will produce oxaloacetate. And then at the same time, this hormone called glucagon, it will inhibit all these steps. But the main thing is that when we have glucose, that will induce insulin. Insulin will go into facilitate what we call glycolysis. Okay, and this metabolism of glucose and of course glucose uptake. When we do not have enough sugar, we are getting into what we call fasting. So for that, we need to make more sugar. So turning from oxaloacetate, we have this enzyme PEPCK. So glucagon is here to stimulate PEPCK to convert these 4-carbon materials known as oxaloacetate, forming into PEP, phosphoenolpyruvate, another form of pyruvate. and then moving backward, fructose 1,6-phosphate, glucose 6-phosphate, and then to glucose. Okay, that would promote this step into here. All right. So it's very important that we know this glucagon would promote this step, and then glucagon would promote these steps. Okay, so the brown, arrows indicating the direction of the promotion are controlled by glucagon. Okay, and then here insulin will stimulate the glucose, turn into G6P, turn into F16BP, and then turn into glycolysis. Okay, so these are the steps that we have to watch. Okay, these are two directions. And after you have understood these directions and their relationship, let's take a look at insulin. Insulin is actually from the pancreatic islet cells. We call it the islet hormones in this slide, showing that this islet means island. So if you have a cross section of the pancreas, where you go, you may go to the wet market. In Hong Kong, the wet market, there are a lot of internal organs from pig, and then you may buy this islet called 猪黃脷. So this is actually the pancreas, and you can see the cross section. So from the cross section, we have this insulin actually secreted from the beta cells. So that is the beta cells here. And then there are also other alpha cells. The alpha cells will secrete glucagon. Okay. And then there are also other cells. Those are exocrine acinar cells. These cells will promote, the structure of the pancreas is actually right next to the duodenum. So the pancreas here, all these exocrine cells would produce enzymes for digestion. So these are called other cells. F cells secrete pancreatic polypeptide for digestion in duodenum. And then these cells also produce huge amount of enzymes being made and then move into the duodenum after the stomach for digestions. Okay. And what else? Yeah, there are also delta cells, smaller amount of cells, delta cells, beta cells, alpha cells, and delta cells. Delta cells would only produce another hormone known as somatostatin. Somatostatin is to inhibit growth hormone. Okay, so these are all the cells we have in the pancreas. There's a cross section. This is what we call an islet. So islet cells, inside there are beta cells, alpha cell produce the glucagon, beta cells produce insulin. Okay, so hyperglycemia, high blood glucose will stimulate beta cells to secrete insulin. So what is insulin? Insulin, what does it do? Increase glucose uptake in cells, in all cells. Convert glucose to glycogen. We call this glycogenesis. Increase amino acid uptake and protein synthesis as well to make muscles or many protein in the cell, and also promote lipogenesis because we want to store the sugar as lipid in our body if we don't use them up. Okay, this insulin could slow down gluconeogenesis and glycogenolysis. That's mainly to counteract the effects of glucagon because we don't need to make more glucose. So obviously that would bring down the blood glucose level, drops into normal levels. Hypoglycemia inhibits release of insulin. Hypoglycemia means the feedback mechanism here when the sugar level drops down to very low level, and then the insulin release would be inhibited. Okay, so these are the functions of insulin. And then if you take, I usually like to do this in a pragmatic way. A day start after breakfast, you have increase of blood sugar that is in brown or red color. And at the same time, serum insulin levels would also increase. I also bet your attention to the different kinds of units, SI units, and this is not SI units, this is a picomole per liter. That is the US standard. They usually use this picomole per liter. Okay, it's up to a couple of hundreds. And then after a meal, and then if you digest the food, then you have a lot of sugar, and then insulin would also come up. Insulin is to facilitate glucose uptake. So the glucose level would drop, and then swing, and then after lunch you have another surge. It also brings about the kind of food that you eat. If it's a starch-rich food or sugar or sucrose-rich food, you can see if you have sugar-rich food, you can see that sucrose is a disaccharide that would make glucose and fructose. Okay, so you can see that the sugar level is higher, and also at the same time the glucose level would high, and at the same time insulin level would high. And then after dinner the same thing happened, and then at night after the normal metabolism we have all the sugar sucked into our body, and then slow down, and then we go to sleep, and then keep doing this cycle. And so that is the diurnal changes of these glucose and also insulin. That's for normal people. Insulin has a very complicated structure because it's comprised of two chains, chain A and chain B. There's another hormone known as IGF, or insulin-like growth factors, IGFs. And then they are actually the same group of proteins with different polypeptide processing. What do they have in common? They all have from the gene leading to the formation of this polypeptide with a signal peptide. And then the B chain, the C chain, C means connecting, and then the A chain, the D chain, and also the E chain or E peptide. All of them, they would release or remove this E peptide. The E peptide will have separate function, which we don't have time to cover in endocrinology. And then this is a signal peptide to guide the whole polypeptide into the ER. And then these are the processing steps. You can see that the connecting peptide is removed, and then only the B chain and the A chains are retained in insulin, with 3 disulfide loops forming a special secondary structure. And then for the IGF-1 or IGF-2, the BCAD chain are maintained together. So these are the BCAD, and then this is the signal peptide is gone, signal peptide is gone, and then this is a B, and then connecting C, and then A, and then D remain intact. Okay. Only in insulin, the two chain, A chain and B chain, are linked together with the connecting peptide removed. Okay, so they have a special secondary structure, and what it does is that it would be stimulated by glucose. So glucose, and there are at least 5 glucose transporters. One of them is called GLUT2. So glucose would be brought in, and then after glucose is in, then the ATP levels will be increased, and then the ATP levels will stimulate those calcium channel, and then bring about calcium signaling to stimulate the release of insulin from beta cells. So this is happening in the beta cells with GLUT2 glucose transporter sensing the amount of glucose from the blood, and then this will stimulate the pathway to release insulin. And what does it do? Insulin would go into the target cells, insulin and insulin receptors. So there are receptors sensing the signal of insulin. So insulin would bind to the receptor and then stimulating formation or promotion of fatty acid from pyruvate, because that will promote lipogenesis. And then at the same time, the signal transduction pathway will stimulate GLU4 to facilitate glucose uptake. So glucose uptake, say like for example in the liver, this is happening in the liver, for the formation or polymerization of glucose into glycogen, known as gonton, in the liver. Okay, so with that we have reviewed insulin and their actions, and the next video we'll go into talking about glucagon. Let's take a break.